Let's begin. A 5-day-old boy born to a 20-year-old woman is brought to the office for evaluation of poor feeding and vomiting. Family history is unremarkable. Physical examination reveals a lethargic newborn with exaggerated deep tendon reflexes and clonus. Further investigation shows that the child is suffered from inherited condition that result in impaired transport of ornithine from cytosol to the mitochondria. Diet restriction of the which of the following substance can improve the patient condition. Before going to the option of discussion, first we know the key things that we give in that scenario. So the child is suffered from deep tendon reflexes, clonus, age of the child, and the transport of ornithine from the cytosol to the mitochondria is not take place. So this thing is necessary for the urea cycle. So which urea cycle is takes place in the liver. So if the urea cycle does not take place, it will lead to the increased level of the ammonia. So it will lead to hyperammonemia. So to solve this scenario, we know the different causes of the hyperammonemia and urea cycle. So the urea cycle is the cycle that convert this toxic ammonia into the urea and which is excreted by the kidney. So the metabolism of the protein, uh, particularly the catabolism, breakdown of the protein is ammonia. Then this ammonia is converted to the urea cycle in the liver. So the urea cycle has a two portion. One portion takes place in the cytoplasm and another portion takes place in the mitochondria. So the carbon dioxide and water react with each other to form the carbonic acid which split to the bicarbonate ion, and this bicarbonate ion is transported to the liver cells. Then this bicarbonate ion react with the ammonia to form the carbamyl phosphate, and this reaction is catabolized by carbamyl phosphate synthase 1. This is basically the rate determining enzyme, and this CPS1 is stimulated by an acetyl glutamate. So another CPS2 enzyme which is present in the cytosol. It is a rate determining enzyme for pyrimidine synthesis. Pyridamine synthesis of the nitrogen bases. So this carbamyl phosphate then react with this ornithine to form the citrulline, and this reaction is catalyzed by ornithine carbamylase. This enzyme, ornithine transcarbamylase, is the most common enzyme disorder of the urea cycle. High yield. Most common urea cycle disorder. One thing. Second is the basically it is a only X-linked recessive. X-linked recessive disorder, while the other enzyme deficiencies are of the urea cycle. Other enzyme deficiency of the urea cycles are autosomal recessives. They are the autosomal recessive. So this ornithine react with carbamyl phosphate converted to the citrulline, and then this citrulline converted to the arginosuccinate. These enzyme is not a high yield, and this arginosuccinate converted to the arginine, and this is again a high yield enzyme arginase which convert the arginine into the urea and ornithine. So this urea goes to the blood and easily excreted through the kidney. In this way, the toxic ammonia is removed through the body, so that these three enzymes are the high yield. which is the rate determining enzyme CPS1, which is present in the mitochondria. So these all the reaction takes place in the mitochondria. This remaining reaction that in the green color takes place in the cytoplasm. So if the urea cycle does not take place, so what happen? So hyperammonemia is basically the increased level of the ammonia in the blood. So the hyperammonemia causes simply divided into the two types: urea cycle disorders and acquired due to the liver diseases. Acquired liver disease, urea cycle, enzyme deficiencies, and ornithine transcarbamylase is the most common enzyme deficiency of the urea cycle. Ornithine transcarbamylase. Another enzyme which is ornithine translocase. This enzyme is responsible for the transfer of this ornithine from the cytoplasm to the mitochondria. So if this enzyme is deficient, the transfer of this ornithine from the cytoplasm to the mitochondria does not place, and the urea cycle does not proceed. In this way, this enzyme deficiency also lead to the increased level of the ammonia. So sign and symptom of the hyperharmonemia is a flapping tremor, slurring speech, vomiting, and cerebral edema. Cerebral edema because of the glutamine. Glutamine induce osmotic shift. So the excess of the ammonia react with alpha ketoglutarate to form the glutamate, and then this glutamate react with this excess ammonia and converted to the glutamine. So normally this glutamate is converted to the GABA. So if the excess of the ammonia is here, so the decrease in alpha ketoglutarate and decrease in GABA. Decrease alpha ketoglutarate lead to the decrease TCA cycle. The treatment of the hyperammonemia is mainly the decrease in the protein diet. Decrease in protein diet, decrease catabolism produce, decrease ammonia. So next is the lactulase. Lactulase basically acidify the GI tract and trap ammoniamine for increased excretion. Next is the rifaximin and neomycin antibiotic. These two antibiotics decrease the production of bacteria that produce ammonia. Next is the benzoate, phenyl acetate, and phenyl butyrate. These are basically the substance that react with glycine. These substances react with glycine and glutamine. React with glycine to form the substance that is excreted renally. So this is basically the hyperammonemia treatment. So the branch chain amino acid like isoleucine, leucine, and valine are normally break down by the branch chain alpha ketoacid dehydrogenase enzyme. If this enzyme is dysfunctional or decreased, it will lead to the maple syrup disease. So the high yield stuff related to the maple syrup disease, in the patient of the maple syrup disease, urine smell like maple syrup or burnt sugar. So the branch chain amino acid diet restricted in the maple syrup disease, and in our clinical scenario there is a no history of the urine smell like maple syrup or burnt sugar. So next, is the fructose. So the fructose is metabolism is the fructose is converted to the fructose 1-phosphate by the enzyme fructokinase. If this enzyme is deficient, it will lead to the essential fructose urea. Fructose appear in the urine. So this is a milder disease. Next, this fructose 1-phosphate is converted to uh, dihydroacetone phosphate and glyceraldehyde through the enzyme aldolase B phosphate. If this enzyme is deficient, it will lead to the hereditary fructose intolerance, super high yield. So the patient of the hereditary fructose intolerance present with symptoms after the consumption of the fruit juice or honey. So there is no clue regarding the consumption of the fruit juice or honey in the clinical scenario. So again discuss the whole metabolism of the fructose. This fructose it's converted to fructose 1-phosphate. If this fructokinase is deficient, the another pathway this fructose converted to the fructose 6-phosphate by enzyme hexokinase. If this is a deficient. So after the fructose 1-phosphate is formed, it is converted to the dihydroacetone phosphate and glyceraldehyde. This glyceraldehyde converted to the glycerol and form glucose in the gluconeogenesis pathway. While this DHAP is utilized in the glycolysis. So the phenylalanine is an amino acid. So the enzyme phenylalanine hydroxylase is the enzyme that converts this phenylalanine to tyrosine. If this enzyme is deficient in a phenylketonuria. So if this enzyme is deficient, the accumulation of the phenylalanine takes place, and this amino acid, this tyrosine become essential for this patient. This is again a high yield. This tyrosine become essential for this patient. The symptom of the phenylketonuria is a symptom and signs. The patient presentation of the phenylalanine, the presentation is microcephalic microcephalic child, hypopigmented skin, and the super high yield stuff, musty body odor. When you see the scenario in the musty body odor, think about the phenylketonuria. So the right option of this clinical scenario is a protein. So in this, basically the ornithine transport from the cytosol to the mitochondria is necessary for the urea cycle. So the protein catabolism produce ammonia, and ammonia is ultimately converted into the urea, which is less toxic substance. This urea cycle takes place in a liver. So in this patient, we restrict the protein diet.